Welcome to the Swine Nutrition Black Belt Podcast, the latest swine nutrition research digested for you. My name is Clayton Chastain, your host for today's episode. Today we have with us Dr. Woongbi Kwon, a recent graduate from the University of Illinois. So Woongbi, would you mind telling us a little bit about yourself?
Yes. My name is Woongbi Kwon. I'm originally from South Korea. I did my undergrad and master in animal science at Konkuk University in Korea. And then I finished my Ph.D. in swine nutrition at University of Illinois under the guidance of Dr. Stein in 2020. My PhD dissertation topic was about branched-chain amino acid metabolism and interactions in growing pigs, and especially we were focusing on the effects of excess leucine in growing pig diets. And now I'm currently working for a nutrition company called Furst-McNess as a swine nutritionist.
Gotcha. So let's talk a little bit about some of that research you did while at Illinois. So I saw a study, like you mentioned, on the tryptophan and leucine levels affecting serotonin metabolism. Could you tell us a little bit about that study?
Sure. Before I started, the whole background about this research is related to the interaction among branched-chain amino acids. And there is interaction also among branched-chain amino acid and tryptophan in growing pigs. So previous study that we've conducted and published in Journal of Animal Science in 2019 indicated that excess leucine has negative impact on growth performance and nitrogen utilization in growing pigs. Interestingly, our group also found that excess leucine reduced brain serotonin concentration in growing pigs. Serotonin is a neurotransmitter that plays an important role in feed intake regulation. The real name of serotonin is 5-hydroxytryptophan. So tryptophan is a precursor for serotonin. There are a lot of information about effects of tryptophan supplementation on feed intake in animals. But in order to be in the brain for making serotonin, absorbed tryptophan needs to compete with branched-chain amino acid because they all share a common transporter called L-type transporter 1 to cross the blood-brain barrier. So if pigs overloaded leucine into the blood, that means the transporter is getting busy transporting leucine into the brain. Therefore, pigs will get a reduced tryptophan uptake into the brain and less serotonin will be synthesized. So our hypothesis of the study was tryptophan supplementation to high leucine diet is needed to prevent drop in serotonin concentration in brain. It is also needed to maintain growth performance of growing pigs. So a total of 144 growing pigs with initial body weight of 28 kilograms was used for this experiment. There were 2 pigs in each pen, and each treatment had 8 replicate pens, so we used 72 pens total. To investigate the effects of increasing levels of dietary tryptophan and leucine, we used crystalline form of L-tryptophan and L-leucine as the source of excess dietary tryptophan and leucine in the diet. We had 3 different levels of tryptophan up to 28% SID tryptophan to lysine ratio, and 3 different levels of leucine all the way up to 300%. Finally, we formulated 9 diets containing 9 different ratios of dietary tryptophan and dietary leucine. So moving on to the results, from the 21 days of growth performance, when we added more leucine in the diet, there was a pretty dramatic reduction in feed intake. As we added more tryptophan, we saw some increase in feed intake, especially in excess leucine condition, but it was not enough to catch up what we have with leucine at the requirement. For average daily gain, there was no surprise. If pigs don't eat, then they don't grow. So we had a similar trend for average daily gain. As we increase leucine above the requirement, we see reduction in average daily gain. When we increase tryptophan above the requirement, we saw some improvement in average daily gain, but it was not enough to catch up what we had with the leucine at the requirement. There was no effects of adding tryptophan if we had leucine at the 100% requirement. But in excess leucine condition, there was a clear effect of adding tryptophan in the diet. And then for brain serotonin, as we expected, if we added more tryptophan in the diets, serotonin concentration in the hypothalamus were increased. But because of competing with leucine on the blood-brain barrier, serotonin concentrations were decreased when we added more leucine in the diet.
Gotcha. So quite a few episodes back, I did a study with Bo Williams, and I talked to you earlier, and you said you saw that one. And it's where he fed excess leucine, but instead of doing it through crystalline amino acids, he did it through extra soybean meal. And then they looked to see if increasing ratios of other branched chain amino acids, isoleucine and valine, and tryptophan increased performance, and it did. And I think at the end of that one, he actually alluded to your study. But do you think it's a similar effect where it's just like a overcrowding of that transporter and like extra competition for leucine and tryptophan, or do you think there could be some additional interaction effects there?
That's a very good question, and K-State Swine Group has been doing a lot of great research related to the branched chain amino acid and tryptophan. And also, like you said, I also watched the Bo Williams previous episode, and that they did great approach to find the right ratios of all four amino acid combination, not only considering each individual amino acid. But unlike our research, they use DDGS as the source of excess leucine, but we use crystalline L-leucine as an excess leucine source. And they also use both soybean meal and crystalline amino acid as a source of additional isoleucine, valine, and tryptophan. That's the difference between those two studies. Like I mentioned earlier, branched chain amino acid and tryptophan share the common transporter, but this transporter is not only for branched chain amino acid and tryptophan. All of large neutral amino acid group, they share this transporter. So in my opinion, if there are many other large neutral amino acid coming from the soybean meal, and they compete with branched chain amino acid and tryptophan, there must be something more on the interaction standpoint. That'd be my answer.
Gotcha. And also what I thought was interesting on your study was the increase in performance when both tryptophan and leucine were increased. Because if it was simply a balance of ratios between the branched chain amino acids kind of competing for that transporter, I guess in my head, I'm no expert on it, but I wouldn't have like hypothesized an increase in performance unless the transporter was originally like not fully saturated. So do you think that maybe the control was deficient, or could you maybe elaborate on that interaction a bit if I'm missing something?
That's a great point. It is possible that the control that we use were, believe it or not, maybe was deficient in those two amino acids. We don't know for sure, but when we check the amino acid composition, it was fine. But especially when we look at the single number on the gross performance table, you could find some, the increases when we added tryptophan and leucine together for the gross performance. That doesn't really make sense. But when we all put together and see the overall trends on the performance graph, increased leucine to lysine ratio had a clear negative impact on average daily gain and average daily feed intake. But there was an interaction between tryptophan and leucine, so increased tryptophan to lysine ratio partially ameliorate the negative effects of high leucine to lysine ratio in this study. But even if we increase SID tryptophan to lysine ratio up to 28%, which is 10% greater than the 2012 NRC requirement for growing pigs, tryptophan supplementation couldn't offset completely the negative effects of excess leucine.
Okay, well, that's all very interesting, and I think that's all the time we have for today. So thank you for coming on the show. And to everyone else, thank you for listening to the Swine Nutrition Black Belt podcast. Please visit us at swinenutritionblackbelt.com. And don't forget to subscribe to our podcast channel so you won't miss out on the next episode. See you next week. Hey everyone, I hope you enjoyed this episode. And we are constantly on the lookout for the latest updates in swine nutrition. And if you have a swine nutrition related research trial that you would be able to share on our podcast, please send an email to nutritionblackbelt@swineit.com, and we would love to talk about your research. See you later.